So as the title suggests, I want to discuss how we get the dynamics from single cell data. First of all, like this acknowledged page, this is really a collaboration with many people. So for today, what I've talked about today, mainly it's by my student Zhang and Xiao from Johnson Weissman Lab, and also Yan is my co-authorized by me and my colleague Weber Har. Then, there are also collaborators I put here. I do not think I'll go to the lifestyle imaging part, but there are also a lot of collaboration and funding. Okay, so let's see, start with the question. The question is, we have been using dynamic system theory to describe a cellular process for a long time. So what I show here is an example cell change from epithelial cell to mesenchymal cell. So this is, we want to describe how this process happens. Well, there are other examples like differentiation, reprogramming. So we want to write down a set of dynamic equations. Let's use a generic example. Let's say we have a system. We know there are two genes. That's important. So identify them. We write down their change over time. So our collaborators, experiment collaborators tell us their say X1, gene 1 have self-activation. Then we can write down a mathematical expression to describe it. As shown here, that really reflects activation, right? Say there's gene 2, there's self-activation, right? Similar thing. Then their mutual inhibition between the two genes, we'll write down expression that describe their repression. So then their degradation. Now we have a set of equations. There are parameters. We can use data help to confine those parameters. So then we got a model. So we can analyze them. That become a lot of tools to analyze those equations. As shown here, there are attractors corresponding to stable phenotypes. Okay, so this is typical procedure for system biology studies. Now the question is, we know the system never as simple as this. There are many genes interact with each other from a highly connected network. So can we, beyond this traditional way of system biology studies, really think about the genome-wide model? So this is a problem that has been puzzling me, really intrigued me since the beginning of my faculty time. I've been thinking about a long time. Especially, I'm inspired by what happens in structural biology studies. So what is shown here is this, suppose you have a protein, you have this crystal structure. Then there's a set of model called the elastic network model. What they say, you got this structure, then you just can write down a very simple set of equations. You represent each amino acid by a bow that connected by some springs. So any bows, they are within a cutoff distance that connect by a spring. You can write down this some energy function, then even can do second order expansion. So you can write down also dynamic equations to do normal mode analysis, those things. It's a very cool model, but works surprisingly well, review a lot of functional relations. So I have been thinking, can we do similar thing for a cellular system? So the beginning of single cell RNA-seq technique have been developed. My lab I started to look at this, but soon we realized it's quite different, challenging. The thing is, for this protein system, the Hamilton, the force matrix H is a symmetric. So if you have a stationary distribution, you can just use the correlation function. You can determine it. So in this model, they either use just constant, spring constant, or they use this called crystal B factors, the thermal fluctuations, to help you determine this gam. But for a general cellular system, it's you do not have this constraint. So this F, for example, even you have linear model, it's not symmetric. And then you need more things, more data to constrain your parameters. So mathematically, let's put in more detail. Suppose we have a system can be described by this set of generic equations, stochastic differential equations. And now you can equally represent by a set of partial differential equations. So in the state space, so this n becomes the number density. So this is for the single cell. This is for population of cells. And then use J and the stratigraphy interpretation is simply given by related to this A convection part, right? A matrix. You have this diffusion, and this term we count for cell division, cell death, that changes. Well, also I here I didn't put this cell-cell interaction, which you can add on also if you have more data. So now you see, if the job is I want to determine those set of equations from data, there are several possible ways. One thing is you have those density function at different time, then you can try to formulate a regression problem. Actually, even early as 2011, I submitted a proposal to NIH, but not surprisingly, didn't really take care of consider it. But nowadays there are several techniques, several methods have been developed, like optimal transport. That's one way. You have a series of this density function. You can try to regress to get this dynamics. But there's a limitation as to this. You need a very pretty high accurate estimation of these density functions, which can be challenging because we talk about multidimensional density function distributions. Another scheme is to do this if you have both the distribution and this coordinate, state coordinate, right? And also it's a velocity, instant velocity. Then you see from here, you can find one way to estimate get what this function form is, right? Numerically, you definitely relax this delta function by some small bin, and you expect all those functions kind of slowly varying compared to this bin size, and you can easily convince yourself that this average quite insensitive to the accuracy of this density function form. Okay, so that's the theoretical mathematical basis. So with this in mind, then comes to it's a few years, about year 2019. Dr. Xiaojie Qiu from Johnson Weissman lab, he pointed me to this paper. So we know when you do single-cell RNA-seq data analysis, we have destroyed cells. So we, in principle, cannot get dynamics, right? But these people said, oh, we got some dynamics within some approximation. Think about the gene expression. You have a transcription, right? Then got the unspliced mRNA, then got to spliced mRNA, eventually be degraded. For those unspliced splice, they're different. There's intron, there's no intron. So you can distinguish them from your data. You can read within certain approximation about this dynamics. You can read, okay, this U and S. Then if there's a certain way, you can estimate this degradation constant, gamma. Now you will get this DSET given by U minus gamma S. So they call this RNA velocity. So after this work, there are extensive effort to try to see how do we get we estimate this RNA velocity better. So even as we talk about this, use metabolic labeling, right? So, but we want to ask a different question. Given you got this RNA velocity, no matter how you get it, can we get more information? If you think about it this way, we have U and S, but let's assume, this S either spliced or unspliced, right? This S is complete for specify the cell state. So then we know then this U also a function of S, right? They definitely assume there's some Markovian dynamics because from mRNA to protein, they're clearly, you may assume there are time delay of things. But if we do not, we only have single cell genome transcriptome data, then let's make the assumption. You can relax there if you have more data. Then you realize this is just a function of S. So then we end up we have a lot of single cell data. We have the expression state and we'll also have velocity. And our biology, with the biology, we have this assumption. They are connected by a set of equations like this. This is a complex nonlinear multidimensional vector function. We do not know what it is, but we assume they are connected by this function, then become a machine learning problem. How can we learn this function from the data? So this is the work really we discussed in this recent publication, and it was mainly done by two very brilliant, capable young people, young researchers, Dr. Xiaojie Qiu from Weizmann Lab, and my former student Yan Zhang, who is currently a Illumina scientist. The idea is also very simple from mathematical perspective. So we do not know what the function is, then we go to a basic strategy we use is go to function space, right? Just for very, in case there are some people who do not know, although I think this is really mass-oriented audience. So the basic idea of this, right? If you have data, then we can do linear fitting. So that's how we do. We try to find the best fit to get this coefficient A1, A0, right? Or they say, okay, this system is nonlinear. Then we get more terms. We try to another fit. Also try to get all those coefficient. We can generalize, right? The basic function can be not just the 1x squared thing. We get the general function, basis function. We try to determine the coefficient. So we're talking about the function space. We determine this. So numerical analysis is called essentially related to the Galerkin method, because we have to truncate, numerically truncate, right? So the idea is you have the data, want to find the function. We have many possible choice of function, want the best fit. So that's what you do is you define a loss function. You want to find that function, give you the best fit with the data. You minimize this loss function. And what you do, your f function, function can be represented the linear combination, some basic functions. Here, for simplicity, we use the Gaussian basic functions. And then we can define a function norm. When you Hilbert space, you have to define a norm, right? This function norm. And then use the loss function, well, we represent get this. So now you just try to minimize this function to determine this coefficient. So the procedure like this, you have all the data, and this is the function form you want to minimize. You minimize this loss function with some regularization. I always try to avoid overfitting. So we use this vector value to take on of regularization, help us to constrain the fitting process. So then just numerical, you got this, and at the end, you got a analytical function form of this vector field because you have the basic functions, you have the coefficients, right? So this is a high dimensional space, and they describe the low dimensional manifold of the data, how the system evolves over this low dimensional manifold. Then you can do further analysis, like I mentioned, this differential geometry analysis, those things. So this is those analysis since published, I won't talk about in detail. Interesting one, the reason I thought about this really because of when I was a postdoc, I read a review paper talking about differential geometry in soft matter physics. Then many years later, sort of applying to this single cell data. So this is quickly go. One example is we got the data from our collaborators. And then they have the data, we can analyzing their vector field, right, shown here. Then we can even project low dimensions. So there we can get even those attractors, repellers, get the attract corresponding to stable phenotypes, can do analysis. Okay, so all those are published, so I won't go into detail. But I rather want to discuss the remaining problem from this work. Besides, we use a lot of assumptions that need to be rechecked and can relax later. There are some other remaining problems. One thing is how can we estimate our velocity more accurately? Since here I mentioned our framework is you give us this discrete value of this R velocity state, we try to find the continuum function form. But how to get one? Especially can we get this dynamic constraints? So it's not just the data. It's data reflecting some dynamical process. We focus on this, the convection part. Can we get full dynamics? We have, when we have the high dimensional model, can we find analyzing the dynamics in this despite there's curse, not curve, curse of dimensionality. Okay. And also, I haven't talked about when we say we project a vector field to low dimension or transform from one representative to another one. How can you do this with vector field, right? Okay, so this is a work we're trying to write up. It's mainly done by my student Zhang, and with some help from Xiaojie on the data analysis. So the idea is this. So we think about all the data sets, the data form a manifold. We always assume that's dynamic process, right? So along some low dimension manifold, the equation, right? Really a mapping, map a data point on the manifold to another point on the manifold, right? Because the constraint, you expect that the velocity should be on the tangent space of the manifold. Notice that the data manifold usually is we have better estimation on this manifold. The velocity is inferred, it may have additional errors or things. So given this, can we use the manifold help to constrain, help to correct the velocity? So you think about the manifold, right? And local neighborhood point in the manifold, we can represent by Euclid space. We can utilize those data points, see this vector replace, the point and the neighbors from this displacement vectors. We can use them as a basis function, basis vector to kind of define this Euclid space. So then we can just project the measure, the raw velocity to this subspace, right? So that's try to satisfy requirement that you need to remove the orthogonal components. So that's the idea. So what you're trying to do, I think, okay, let's just, okay, I have this velocity vector, raw velocity vector. I try to find a component projected on this subspace. We define loss function to minimize. The idea is very simple. But you think more about it, then you realize some problem. The problem is one thing, our base function is non-orthogonal, that's okay, but redundant. Then because redundant, there's an infinite number of possible combinations. You have undetermined factor, right? For you to kind of form this decomposition. But more severe is this. So because I have a redundant basis function, basis vector, right? And this is not just a perfect approximation of this manifold. Most likely, you will have some small component along this orthogonal subspace. So then, when you try to do this, because of redundancy, because this residual component in the orthogonal space, then you see the system, optimize. They always try to utilize this, right? Have large components for this vector, right? Suppose there are data that have components on this orthogonal subspace to give the very high component of this vector displacement, then utilize remaining ones still can satisfy this relation. So you try to project onto this subspace, but fail. To do this, we try different way. Then eventually we realize we need provide some guidance to this loss function. So what we do, we borrow in the field before previously. People also trying to do that, but there are no, they propose a very heuristic method, but it's no real mathematical foundation. The argument is, okay, you can think about this velocity, right? If this velocity is along with the displacements, then most likely there are high chance this cell state will jump to this one. So if on the reverse, right, it's unlikely. So use their correlation kind of heuristic define a transition matrix, but there are no mathematical foundation, right? But we utilize help us to guide this choice of this thing. So that's what we end up. So with this, so we got this correction. We try to help to correct this velocity onto this tangent space. Now we ask question of the dynamic equation. I mentioned the genetics equation, and we have this corresponding PDE, and the F function from the dynamo. That's our paper. We already found a way we can estimate. How can we get the full dynamics, right? How can we analyze? How can we project and do coordinate transformation? So we realize we can solve this. One way to do it by doing in a discrete representation. So this is related to a work I did many years ago when I still a postdoc. Suppose you have a PDE. How we solve it? You have discrete grid points. You do represent by central difference or something. Another way you do it, you divide your space into some different regions. Then you can this equation can be represented as a transition flux exchange between different regions. You can discretize this continuum equation by some Markov jumps between neighbors. So this is a work I did before, but there's a problem, right? Because if you work on high dimension, so each dimension you have n points, then quickly, right, the 2-dimensional n square, 3-dimensional n cube quickly goes up. So how can we avoid this curse of dimensionality? So you realize, the data really on low dimension manifold. So if we discretize along this manifold, even so there's a manifold embedded in the high dimension space, but we only need to discrete on the low dimension manifolds. That kind of bypass this problem. So the way to do is you can discretize your data, use your data points. That's a natural discretization of your manifold, right? So you can convert your transition dynamic as a transitions, jumps among these data points, right? So you can assume this data, right? You can jump exchange with the jump to the neighbor points, so you can make a transition. So this one, again, for using this discrete graph in this. So when we talk about this Euclid, the manifold embedded in the Euclid space, right? So when we need to talk about the vector. The vector have dimension, have rotation, have this weight, length, and also have this orientation. But when we talk about the graph, then the orientation is really irrelevant, right? What I care about is really the connectivity and its weight. And this corresponding, really, because those, this geometry information really irrelevant for us to describe the dynamics. So we only care about the topological relations and some dynamics about manifold. So this corresponding, this I talk about here, you all know, right? You have, you can, topological equivalent shapes, you can convert them, transfer them without changing a lot of topological features. So with this, what do we do there? You have a network, discrete network. There are a lot of efforts have tried to define this discrete vector calculus on finite directed networks. So there's, for example, there's node I show here. So what do you do it for continuum? We have this, let's say the scalar potential of the gradient. Well, that's corresponding on graph is given by, you see the nodes, right? You have the nodes, you have this edge, and then so this edge, by the way, this edge is just an abstraction of this displacement vector I just mentioned. Then you can also define the gradient, what correspond to this. And for vector field, then you can also define something like this. So this is this definition will help us to satisfy like the Green-Stokes relations. Okay, so this is a well studied in the field. So we utilize this. So with this, now you see, I have a dynamic equation. Now I represent on a discrete graph. And also I also already have a discrete representation of my vector field, right? So now it's just straightforward for us to translate this continuum PDE on is a transition model in a discrete graph. Okay, you got all those things. So mathematically, it's corresponding to this. So you have your manifold in this space, right? So you discretize your manifold into different regions. Then we are talking about right? Each point represents a region, right? As volume in this manifold. Then each neighbor volume, they can exchange the flux. So that's really this one tells us. So you see clearly, how then I mentioned this gauge choice, right? This corresponding how you choose your manifold. You choose your sub-regions, then you get a different weight of things. But the point is, once you choose them, then this boundary of things are fixed, right? If you do a distortion of things, but still, if the point belong to a sub-region, it's always belongs to sub-region, right? So then also the number of particles in this region, the number of the flux past the boundary, all those are invariant to how you deform your region, represent, right? This coordinate transformation. So this corresponds to a very basic concept in physics called the invariance. That's we want the dynamic equation to be invariant, right, to how you choose the coordinate system, like the F equals ma, right? So for in the framework of Newton's dynamics, not the relativity. So then we know mass to be independent of how you define your coordinate, while A and acceleration and force should be covariant and using this Galileo transformation, right? And now you see this has become very straightforward for the transform vector field, the dynamic equations between different representations, because all those numbers of the flux should be invariant. That's why it's so convenient if we look at the model, that discrete model. So you have this representation, suppose you have this representation, and you can define your vector field by this and displacement vector things. And then if you transform to another representation, you distort all those regions, but those relations still hold, right? It's invariant. And all can be mapped to this corresponding, the same graphs, okay? So this is a simulation. You show this is a two model. One is two genes regulate each other. There's multiple genes. So you can see this correlation kernel is what I mentioned, this heuristic model, which do not have really mathematical foundation. So it won't, can provide some qualitatively the direction of things, but quantitative the relation are lost, right? But with a new way we do this, really try to represent the vectors on the graph, then we can constrain, we can really preserve all those quantitative information. And also we can really to describe the dynamics, like so systems start from here, propagate. Then with time, we can really recapitulate all those dynamic information I show here. There are certain parameters, like here I show, for example, the diffusion constant. I think in this simulation, my student just use a constant D. It shows, okay, arrange in environment. But definitely, if you have time series data, you can constrain them. Yeah. So that's my, I think I'll summarize my final. So this is, I want to show you, so, now we have a lot of big data, right? People talk about this. There's emerging trend to analyzing data in the framework of dynamic systems. And I think there's still a lot of challenge, but this is a very promising field. I really think this field will advance very quickly in the next few years. Okay, I think I'll stop here.